Hey guys, so my name is Azaz Chaudhry and I'm a part of Group 1A. And so to understand Group 1A's paper, we must have an understanding of B cells and T cells, but we cannot do so without also having an understanding of stem cells. So hopefully by the end of this presentation, these subjects will be a little more clear because we will have introduced stem cells, we will have identified B cells and T cells and their receptors, we will have classified different T cells, and we will have described how B cells and T cells function. So, what makes stem cells so important? Well, stem cells are unique from all other cells because of three specific characteristics that stem cells share. First, they're capable of dividing and renewing for long periods of time. This is known as proliferation. Two, they're able to proliferate just as nonspecific stem cells, meaning they don't become other specific kinds of cells. And three, they're able to become specific to tissue or organs with special functions if they are under certain physiological or experimental conditions. So as you can see from the image on the right, the stem cells can become either nerve cells or liver cells or cardiac cells or even blood cells. And so this is known as differentiation. And so in the bone marrow, when let's say a stem cell decides to differentiate into a hematopoietic or hemopoietic cell, it becomes a blood cell. And so an important thing to know is once it becomes a blood cell, it cannot differentiate into a different kind of specific cell. So once a stem cell has become a hemopoietic cell, it cannot become, let's say, a neuronal cell. And so just as there are different ways for a stem cell to proliferate or differentiate, there are also two main different kinds of stem cells. And so the first kind of stem cell is the embryonic stem cell. And then this is present in the developing human embryo and all other mammalian animals for that matter. And then the other kind of stem cell is the somatic or adult stem cell. So these two stem cells are different in the way that they're able to divide and differentiate. So two terms that we need to understand in order to understand these differences are pluripotent and multipotent. And so embryonic stem cells are said to be pluripotent, meaning they can differentiate into any cell type that makes up the body. Whereas somatic stem cells are multipotent, meaning they can become one of many different types of cells, but they're more specific than pluripotent cells. And so, just to show for the example of this pluripotent versus multipotent aspect, embryonic stem cells can proliferate in vitro for up to a year, whereas somatic cells cannot. And so the image on the right shows if we start out with the hematopoietic cell that I mentioned in the previous slide, it can become all these different types of cells. And so a stem cell becoming a hematopoietic cell undergoes a process called hematopoiesis, which is just the differentiation of a stem cell to form different kinds of blood cells, more specifically either red blood cells or lymphocytes. And so lymphocytes are one of the five main types of white blood cells, the other four being neutrophils, monocytes, eosinophils, and basophils. We're going to focus on lymphocytes because those consist of the B cells, T cells, and natural killer cells. And so lymphocytes are found in the lymphatic system, obviously. They are the main consistency of the lymph, which is what flows in the lymphatic system, and they're critical for the immune system. And so the immune system functions in two ways, either as an innate response or as an adaptive response. And so the innate response is what the natural killer cells are for. We're not really going to talk about those in this presentation, but innate responses, the natural killer cells follow the innate response system, and that's pretty much it for that part. And then so the B cells and T cells, however, follow the adaptive immune response system. So this basically means they become active after exposure to an antigen, either as an effector or a memory lymphocyte, which we will cover later. So let's start with B cells. B cells are also known as B lymphocytes, and they get their name because they're produced in the bone marrow. So as you can see, B for bone marrow gives you B lymphocytes. And they're particular for specific antigens, and their receptors are known as B cell receptors or BCRs. On the other hand, we have T cells, which also, even though they are also produced in the bone marrow, they mature in the thymus, which is an organ in the lymphoid system. So again, the T for thymus gives you T cells. But also like B cells, they bind specific antigens and have T cell receptors. So now we're going to talk about how they both are similar. So BCRs and TCRs are both integral membrane proteins, meaning they span the plasma membrane and have systolic and extracellular domains. They're adaptive, so they are produced before interacting with an antigen and modify themselves after they interact with said antigen. and they both have unique binding sites for each antigen. So where they bind on the antigen is known on the epitope, and they bind non-covalently. And so both of these are made by the genes which are assembled by recombination of DNA, which is which gives you the adaptive response. There are slight differences. The structure of BCRs and TCRs are different, and the genes that obviously encode for them and the types of epitopes that they would bind are different. So how do B cells function? B cells bind to intact molecules, meaning they either bind one, soluble molecules present in the extracellular matrix, or two, they pluck cells off of other cells that they identify as a foreign antigen. So once they bind the antigen, they engulf it via receptor-mediated endocytosis, meaning they absorb it into their own plasma membrane. And so once the antigen is digested, it's digested into fragments, and then after this digestion, it is displaced back onto the cell surface via class 2 histocompatibility molecules, which is also known as MHC2 complex. So T cells come into play here. So there's a couple of different types of T cells, and the first one we'll talk about are helper T cells. So helper T cells are specific for an antigen in the MHC2 complex, and they secrete lymphokines, which stimulate B cells to enter the cell cycle and develop in order to clone their BCRs that have bound this antigen. So helper T cells have to function in conjunction with the B cells. The other kinds of T cells we have are separated by the kind of glycoproteins found on their surface. So the first one we'll talk about is the CD8 T cells. And the key characteristic of CD8 T cells, besides their CD8 glycoprotein, is that they function like the B cells had an MHC2 complex, T cells use the MHC1 complex. And so CD8 cells function by secreting a cytotoxin, and so they secrete this toxin that destroys cells to which they are bound. And CD8 cells monitor all cells in the body and destroy anything that expresses foreign antigen fragments in the MHC1 complex. The second type of T cell is the CD4 plus cell T cell, which like the B cell uses the MHC2 complex to bind antigens. But the CD4 T cells either function either through a cell-mediated or an antibody-mediated response. So the cell-mediated response is the CD4 binds the antigen, and then it releases lymphokines to attract other cells to the area, which causes inflammation. So whenever you have inflammation in your body, this is because of CD4 cells. And so the accumulation of cells that make the inflammation attempt to wall off and destroy the antigen. So that's known as the cell-mediated response. The antibody-mediated response is when the CD4 cells act as the helper T cells. So they respond to the the B cell binding to the antigen in the MHC2 complex, and this leads to the development and the activation and the cloning of the proper cells to deal with this antigen. So once one B cell and CD4 helper T cell has bound to an antigen that started, the lymphokines have been released to cause the cloning of other B cells. Other B cells will be in your body just specific for this antigen in case it's found in your body again. But in order for the helper T cells to function, they have to be activated. And to be activated, they have to have a TCR with the MHC2 complex. They must bind to a class 2 molecule, and they must also bind to co-stimulatory molecules, meaning the B cells. And after this binding and the presence of the TCR with the MHC2 complex, then the helper T cell is fully activated, which leads to the stimulation of the cell cycle and the secretion of lymphokines. Hopefully this talk gave you guys a better understanding of B cells, T cells, and how they relate to stem cells, and to further apply it to Group 1A's paper. Thank you.